All right, it's nice to welcome you to Chapter 4. We're moving right along. We'll get through this particular chapter hopefully in just one session, maybe two, we'll see. Uh, I'm not worried about that. I just want you to have the material as you need it. So we're gonna talk about the cell. Hopefully, I've had a chance to look at some additional videos that I placed. I'm going to include some here, and depending on time, we'll see how far we get. But uh, that should change your perception of what the human cell is about. It really has not done you well if you've only kind of reviewed a 2D, kind of just a general discussion of a cell, which is pretty typical. Hopefully, I can break that mold today a little bit. Describe the structure and function of the cell membranes. They're so important. It's like the universal shopping bag. It's so useful. The cell uses it for many functions, and I want to describe some of them. Molecules move across these membranes. It's sort of like a semi-permeable type of deal, where it allows things in at once and doesn't allow things once it's modified it to leave. Uh, it just makes really good sense. There is the analogy about the functions of a cell. I've actually written a summary on that, the analogy of the cell, which is available for you. And a lot of times students like the business analogy. It kind of puts things that you know and relate to in terms of maybe what you don't know so much on the cell, and it helps, and the students recall it a lot better. So let me know if that helps you. That way I can continue to refine it if it's important to you or not. 9 important landmarks in eukaryotic cells, which are just kind of the structure function. So we know that everything is made up of cells. I mean, there's not too much argument there, I would imagine. But the diversity of these and how these cells are organized, it's huge. It's diversity in things that are very large, very small. It could be made up of single cell, like my friends in bacteria, to multiple hundreds of trillions of cells, as like a human. So it's quite amazing. So the cell theory, of course, is the underlying or unifying theory in biology. Everyone accepts it, and I think it's pretty obvious that everything is made up of cells, and all cells arise from other pre-existing cells, and there was a huge era in science history of spontaneous generation where life just boom appeared, but that kind of was dismissed because of the microscope. So we'll talk about that. Ostrich eggs weighing more than 3 pounds each. Wow. Sturgeon eggs, $700 per ounce. Human eggs, thousands of dollars per egg. Wow. It's interesting that it's uniform in terms of how these things exist to some degree. The basic unit of any organism is the cell. I mean, it goes without saying. They can function independently and perform all the necessary functions of life, including its own reproduction. All living organisms are made up of one or more cells, and all cells arise from other pre-existing cells. The only thing that kind of leaves that definition sort of scrapped is viruses, and we'll talk about viruses. They are living organisms. It's just more of a really simplified, like a flying gas can, as they had in the military, where they stripped down everything on an aircraft to make travel farther and that sort of thing. Well, it's similar to that. It's infectious DNA or RNA that really what we're talking about with viruses. So eukaryotic cell, remember those are the more advanced than the prokaryotes. The eukaryotes have a central control structure with a nucleus. It has a guarded environment. You just can't walk into the CEO's office. There's a dual membrane that kind of keeps that contained in the nucleus. Prokaryotes don't have that. It's sort of like a floating rubber band in the milieu or the the jelly stuff inside a bacteria cell, and it doesn't have any sort of protection in that. But anyhow, that's one of the big differences. So prokaryotes are sort of your bacteria, and these have there's your rubber band, sort of say, floating around, and these smaller ones are referred to as plasmids. These are sort of a subset, usually conferring antibiotic resistance or other characteristics that the cell can share with other bacteria, and these are sort of disposable because they can live without them, but it certainly helps them within an environment. And you can see all sorts of structures, but it's a single cell. It has a flagella that actually spins. The interaction here is actually a motor, and I'm going to talk about that at some point. These are adherence mechanisms, urinary tract infections and things like that. The pili adhere to the epithelial cells and cause disease. The capsule is sort of a protective seal around the bacteria. It's still permeable. The cell wall gives it a little bit more of a shape, and it's more restrictive as what gets in and gets out of the cell. So all I can tell you is a prokaryote in this case here is a lean, mean fighting machine. It's quite amazing. Every cell on Earth is either a eukaryote or a prokaryote, and that's a big deal. And so the prokaryotes have no nucleus. As I mentioned, it's just a circular piece of DNA, and they're a single cell. That's how they hang. They're just single. They don't. They do interact with other cells and communicate, but it's not anything like eukaryotes. And they do have huge metabolic diversity. In other words, they utilize and secrete all sorts of different chemicals depending on the organism. So there's several videos that I've posted in this section, and these are there for you to give you a different perspective of what's going on inside eukaryotic cells. They're well done. They are not cartoons. They are drawings of the atomic view accuracy. It was actually, these were done at the atomic level using X-ray crystallography, and they've been able to photograph using amazing technology. This was done at Harvard. Raymond Liu is a gentleman that was the technical consultant at Harvard that wanted to convey what actually goes on inside a cell to students, and this is sort of setting up a new standard. So if you get a chance to view these, I think I have some here. Let me pop over here, and let's not do that one right now. Let's go to the inner life of the cell and do this one, and just go to that web link. This is what you'll see. And then again, this was done by a company up in Baltimore called XVIVO. They did it as a research project with the BioVisions of Harvard, and so the inner life of the cell is quite amazing. You can see the interactions of various components, pieces, and parts. Communications between cells are really just structural proteins talking, causing shape change. That's how we communicate from cell to cell. A raft floating in the phospholipid bilayer. This is a sort of like putting a painting on a wall. Those are ways to attach the painting to take up more surface area so it holds it. Very similar. The infrastructure inside the cell keeps it from getting smashed. Roadways to transmit various or to move things from one area to another, spontaneously form and break down. We have enzymes that break and cut proteins, and so these pathways build and subunits come together spontaneously and break down spontaneously. There is a signal. Here's a kinesion. This is the federal FedEx delivery guy. It goes around the cell and delivering packets of freshly made proteins anywhere it's needed. and these little delivery guys carry a pack, sort of like an ant, 1,000 times even more. Ribosomes producing these messenger RNAs. Well, they're riding on top, so you can see the ribosome now getting together, forming a protein based on the sequence of that RNA, messenger RNA, and making all the proteins inside the cells. It's quite amazing. Here's the docking of ribosome that made a new protein and delivered it on inside of that particular organelle. Those are the sacs that the kinesion carries. It's phospholipid bilayers that come from the shipping department or the Golgi apparatus that have been freshly packed with new made proteins and various things. Then that backpack will actually go and meld with the membrane, and it contains all the proteins that were made that may be surface related, like this, for communication. There's a raft forming with cholesterol that keeps certain similar functioning proteins together. There's a monocyte that's changing its shape, and it's going to actually skinny in between cells. It's part of the immune system, and cytokinesis is going in, and these cells go around and policing all the different areas, making sure everything is working properly. So I hope this gives you a sort of a really quick bird's eye view of what's going on. I have other videos that you may want to review. It's just amazing what Michael Astrakan at XVIVO has done. These are the videos that he worked, and again, this just shows the compactness of what goes on inside a cell, and there's a lot of spaces that just aren't there. It's pretty packed, and it's amazing that we get functioning. So the point here is, I say off the top as you watch this, if you don't drink water and you start to get dehydrated, that space decreases even more, and so you can see why certain systems don't work. See, these are just proteins inside cells that are doing whatever it is that they're doing. Now the shaking is the Brownian movement of the water, just with temperature 98.6. So things are kind of agitated, moving around, and you can see there's a lot of proteins being made, and ones that are doing whatever it is that they're supposed to be doing, and it's just constantly going on. And it's just amazing to think about. This is going on in the 100 trillion cells in every cell in our body. It's quite amazing. And so you can look through these videos that really give you an amazing view. There's the kinesion attaching using its feet. A slightly different portrayal, but doing the same thing, and going on to the active molecule, moving down and carrying its membranes. A slightly different type of drawing. It is accurate, though. It is representing what is out there. So again, this was just something I wanted you to view, to give you the impression, the better impression what's going on inside a cell. And so I have the one you were just watching, and you can take my slide set and copy and paste, or just click on the graphic. It will take you there. I also have one on the mitochondria and how it works. These are nanomachines. I'm going to spend some more time talking about these later on, but it won't hurt for you to view the video. It's quite interesting. These spin, and what drives this is a sort of a battery effect across this membrane, and we're going to talk about energy and that sort of thing. But the energy actually that drives this is hydrogen, and it's the hydrogen from H2O, and we break down hydrogen, and then of course you get hydroxide group, the OH group, and it's the relationship of those two, and pH and all that. But it's slightly positive charge as the hydrogen goes from one side of the membrane to the other. and that causes this to rotate, because these, you see the bright areas, are slightly attracted to that positive charge, and as a result it starts to spin, and so these spin, and the movie shows that. You can see this is it shows you how ATP synthetases actually spin, and then up at the top when it's spinning you have a stator part that forces ATP or ADP to get in close to some association with inorganic phosphate to make ATP. And so these are the energy power plants that are making ATP that are floating around that actually fuel the cell to do all the things that we do. So it's the subunit. One subunit contains the ADP, and the other subunit, it's very close location, it contains the inorganic phosphate. And this structure, because it's spinning, puts those two components so close together it forms the ATP. And this is a machine. It's really, there's nothing else more you could say about it. It's a machine. So I hope that series of videos will kind of give you a different view of what's going on inside a cell and how phenomenal that relationship is. And then cells can do all sorts of things. It's amazing. Porcupines. I don't know how many quills I pulled out of dogs' mouths from not understanding that's a defense mechanism. Slime molds to plants that have all sorts of interesting aspects. And so eukaryotic cells have the organelles and those specialized features. Now, the bacteria don't have a lot of the cubicles or the special rooms, but believe it or not, they have a lot of the same nanomachines that we have, and they function in very similar ways. So don't think the bacteria do things that are sort of like, old-fashioned or anything. It's just that they lack the all the specialized rooms, but they still kind of do the same nanomechanical type of manufacturing that goes on. So I hope this section here you followed me on that. So eukaryotic cells are about 10,000 times larger than prokaryotes, and that's because you got to take up and consider all the compartments or the cubicles that you're going to be putting various structures in and that sort of thing. You can see it under a microscope rather easily, and I'll show you some slides that's doing that. And many of the structures inside these are called organelles because they function sort of like a specialized uniform feature or function, and like an organ, and that's what they do. So here's just a drawings that you're probably more familiar with. A 2D drawing, kind of boring. It doesn't show much, but now that you've seen what actually happens inside of the cell, I hope it gives you more of a view and an appreciation of these 2D drawings, because it's not really like that. These are those dynamic. If you remember the ribosome docking in a protein, and it was the ribosome would actually go across and form a protein inside the membrane. It kind of injects it in there like a 3D printer does. Anyhow, so just looking through the names, we have the nucleus, which has sort of like the analogy I have is the CEO's office. You can see the dual membrane, and these are sort of hatches that allow certain functions through, or proteins or whatever through. Also, messenger RNA is manufactured inside here and come out these ports. And then you have really close to that is we have the manufacturing floor, and you can see it's studded with these little ribosomes that are embedded in these membranes, and that's why we call it the rough endoplasmic reticulum. Now, all endoplasmic reticulum is a fancy word, but just means a series of rooms or caverns, and that's what it is. I mean, it's just a series of these that have the ribosomes on here, and it's sort of like a shop manufacturing floor where sparks are flying and people walking around in their work clothes, and it's quite a lot of activity because it's manufacturing all the various proteins it needs, from the messenger RNA, which came from the brain trust, of course, of the never to be touched DNA that's in here. We make copies of the DNA to make forms of RNA, which now can be read out on the manufacturing floor to make proteins. And that's what you're looking at. And eventually, we need to move whatever's being manufactured, just like regular in the analogy of the cell. It goes to the shipping department, and this is the sort of the Golgi apparatus, which is the shipping department. So the back door allows things to come in, and then the front door is where you see the kinesians taking out these, moving them to various others. Now, you might wonder, what are these pipes over here? Well, these are the smooth endoplasmic reticulum portions. And yes, there's proteins that go directly to the shipping department, but there are some that go through further processing, or we also detox cells, detoxify using the smooth ER. These are smooth because they don't have the ribosomes bound to them. So there's other things that are going on, and they too are shipped. It may go back through the network or go directly to the Golgi apparatus or the shipping department. And then the generators inside the cell are, of course, the mitochondria, and those are the ones that are studded with that that power wheel I was showing you before, the ATP synthase that spin. And there's just billions and billions of these. And if you notice, it's a bag within a bag. Really, if I were to show you a battery, it works the same way. So what we're doing is we're transporting hydrogens across there, so you get a higher concentration of hydrogens on the inside of this bag, and that has now a battery effect across that membrane and spins those ATP synthases to make ATP, which is what drives all the functions of the cell. So think of it, the mitochondria as the generators that are roaming around, and if they need more power, they get more mitochondria or more generators rolled to them. And it's really interesting. Plant cells, very similar. If you look at the structure, has pretty much all the same types of infrastructure, but the building has to be kind of beefed up in terms of the analogy, because of course we're outside in the weather getting beaten up, so we have to have sort of a thicker cell wall. And then because we store water and various things that are unique functions of a cell for a plant, because the water is really, really important, because it's water that plants use, the photosynthesis. We actually crack the water. We're going to talk about this in the energy, and we use the water as the source of energy. So these are like hydrogen-driven machines, these plants. Now, you say, well, Dr. V, what are you talking about? Well, the water in the plant is actually broken down into hydrogen and hydroxide, and it's the hydrogen that's used to charge batteries, a battery effect in the chloroplast. And so these are sort of like solar cells inside a cell, because it's deriving its energy from the use of water, using sunlight to vibrate the water so violently that it pops the hydrogens off, and then that's what drives the system. And I'm going to go into detail about this if you don't believe me. And that's truly what's going on in photosynthesis. So I will share that with you when that time comes. So plants have a rigid cell wall, as do fungi and many of the other proteins. The proper way to say fungi is not fungi. That's somebody you invite to a party. It's fungi and many proteins. The chloroplast, also found in some proteins. Plants also have a vacuole, large center chamber, occasionally found in animal cells, for different reasons. They store fat in animal cells, but water and various other things that plants use are stored in these containers. Endosymbiosis theory is very, very interesting. This is rather complex, but I don't know, it seems like nature utilizes the theory that if don't reinvent the wheel. If something's out there, bring it in. And it's what they did, well, if you think of it in a theological sense, that we have certain organelles in our system that actually were free-living organisms. And they kind of joined into existence with our cells. And so the mitochondria is one such thing. It actually is more like a prokaryote, because it's genetic material that's inside the mitochondria is unlike our DNA in the human cell. It's circular, just like protease. And chloroplast, same thing. It seems to be at one time a free living, and it's come in and it's taken up shop inside plants. So, but endosymbiosis, the idea is that we have prokaryotes that have partnered with our cells to carry on two really, really important functions, that of making energy, either in regular cells with the mitochondria, and we're going to talk about how we extract that energy, and chloroplasts using sunlight to crack water to drive those ATP synthase motors, and we'll talk about that. But there you go. That's really what's going on, and over time I will convey that to you so that you feel like you've really got your arms around it. So this is more of a pictorial, just showing the endosymbiosis theory, the ancestral prokaryotes coming in inside the cell and taking up shop and providing all the features and necessary things to make the life sustainable by making ATP. And that's the theory, and so far it seems to be holding water. So endosymbiosis, the theory of this is kind of broken up. You can see it's just a review, and you can read through that, but it's all in how it works together. But the real proof of the pudding is the fact that it's circular DNA in both the chloroplast and the mitochondria that are like prokaryotes, not like our DNA. And the mitochondria, oddly enough, come from the genetics of the mother, and it's quite interesting. We'll get into that later on. And so it's just another way of representing the endosymbiosis in summary. A lot of students have trouble with this. It's like, well, why are two different life forms coming together? I mean, that's kind of icky. I mean, it sounds like we're contaminated or infected or something. And it's not really like that. It's a matching of molecular tools is the way I like to look at it. And so anyhow, so there's enough graphics here in my slide set here that you can kind of follow the idea. We are visual learners at Wake Tech. It seems like 80% of us, I've done enough testing over the years, it's 80%. So my guess is a high probability that you're a visual learner, so I try to throw in a lot of these graphics to help you with that. And the key points, the summary of endosymbiosis, you can see again, it's just restating everything that we've talked about. And the bigger picture summary, the eukaryotes are single-celled or multicellular organisms consisting that contain linear strands of genetic material, chromosomes, that are usually inside a nucleus. And the eukaryotes have organelles, or the rooms, or the cubicles throughout their cytoplasm. Now they're larger than the prokaryotes, but they carry on a lot of particular functions. So the rest of the slides I have here step us through the various aspects of the eukaryote. So I think just for decompression and to give you a chance to review those videos, I'm going to stop here, call this part 1, and then continue on and conclude this chapter 4 with going through the various different types of structures inside a cell, and relating the movies that you have here with the inner life of the cell to help really convey it. And I would use the analogy of the cell to help follow along, to give you that relatedness of functions within a business or something that most folks can relate to as to what's going on. So as I say, I'm going to stop here. I'm going to make that decision because I'll give you a chance to view the videos if you haven't, and then we'll pick up right here next time. Thank you. Be safe.